Hello everybody, and welcome to mini lecture number 2 for this module. This is the cell cycle and mitosis. Again, you want to take notes on this material because we'll use it over time. So please, if you need to pause here, go grab your notebook, something to write with, and I'll see you right back. Otherwise, let's get into it. In eukaryotes, the genome consists of several double-stranded linear DNA molecules. Each species of eukaryotes has a characteristic number of chromosomes in the nuclei of its cells. For example, humans have 46 chromosomes and fruit flies have 8. In humans, somatic cells, which are cells of the body, have 46 chromosomes in 23 pairs. Human gametes, or the sperm and the egg, have 23 chromosomes each. So we can see here in this karyotype, this is what's here, it's just essentially a picture of the chromosomes of an organism. We can see in this karyotype the 23 pairs of chromosomes matched up together, and we call these homologous pairs. So when chromosome matches together, homologous, so same chromosomes together. A typical somatic cell contains 2 matched or homologous sets of chromosomes, one set from each biological parent, which is called diploid, and we can see that term here. Diploid is 2N, and N means sets of chromosomes. So diploid cells have 2 sets of chromosomes. Gametes are designated as N, or haploid, because they contain only one set of chromosomes. We'll look more closely at gametes next week when we discuss meiosis, but for this week we'll focus in on somatic cells. Each copy of a homologous pair of chromosomes originates from a different parent. Therefore, the different genes or alleles themselves are not identical, although they code for the same traits, such as hair color. The variation of individuals within a species is due to the specific combination of the genes inherited from each parent. Even a slightly altered sequence of nucleotides within a gene can result in an alternative trait. So here, this is my husband, this is me, these are our 2 daughters, okay? You can see that each daughter looks very much like one of her parents. The skin tones match up almost perfectly, but my youngest daughter has my smile and my hair texture. And in baby pictures, this is exactly my face. My face looked exactly like that when I was little. My oldest daughter actually looked like a fairer skinned version of my husband's face for the first 2 years of her life. And they still have, you can't tell really here, but they still have the same face shape. Now, before we get into division, let's start by looking at some basic chromosome structure. Okay, and now I'm going to make you endure my drawing. So here we go. A chromatid is a single unit of chromosome structure. Looks like this. That's a little structure right there. All right. A somatic cell's chromosomes are made through the duplication of chromatids. Okay, so this is one chromatid during cell replication, which we'll talk about when we look at mitosis. Then this one chromatid would be duplicated. Whoops. To give us this kind of traditional chromosome X structure that we see. Okay, so chromatid, sister chromatids, one chromosome. Now we know that each cell contains a chromatid from each parent. We just saw that in the karyotype. So those would both be duplicated, creating what we call homologous chromosomes. Those are 2 chromosomes from the same chromatids. So they have this, they code for the same traits, but they're, one from each parent. Homologous chromosomes are here. Each chromosome is made of sister chromatids. This is an individual chromatid. Chromosomes are connected by structures called centromeres, which I've scribbled here in black. And during mitosis, these structures called kinetochores, which I'll just make right here, they kind of bracket each end of the centromere. So during mitosis, these kinetochores develop at the centromere, and then the spindle fibers will attach there and pull the chromatids toward the pole. That might not make sense now, but it will be in just when we get to the mitosis part of this module. Chromosomes are only visible and distinguishable from one another when the cell is getting ready to divide. Now, just for illustrative purposes, if the DNA from all 46 chromosomes in a human cell nucleus were laid out end to end, it would measure approximately 2 meters. So that's longer than your arm stretched from tip to tip. However, its diameter, so the width of it, would only be 2 nanometers, which is extremely tiny. So considering that the size of a typical human cell is 10 micrometers, which is also tiny, DNA must be tightly packed in order to fit in the cell's nucleus. So not only does it need to fit in this 10 micrometer cell, it needs to fit in a smaller space, which is in the nucleus inside the cell. So at the same time, it needs to be compacted enough to squeeze into the nucleus. It has to also be readily, pieces of the DNA sequence have to be accessible for gene expression, which we'll talk about in detail in 2 weeks. So for this reason, the long strands of DNA are condensed into compact chromosome structures during certain stages of the cell cycle. And there are a number of ways that chromosomes are compacted, and it's kind of another part of the cell cycle is just a compaction of chromosome. So in the first level of compaction, short stretches of DNA, so we see our DNA double helix is this purple structure. Short stretches of the DNA are wound around these histone proteins, which are the blue circles here. So you'll have a stretch of DNA wound around the histones, and then this pink thing is called linker DNA, and that's just the kind of the space that goes from one DNA piece wrapped around a histone to the next piece of DNA wrapped around the next histone, linker DNA. 8 of these histone DNA, this histone DNA complex together, when you have histone DNA wrapped around histones, we call that chromatin. Okay, that's that compacted structure is chromatin. Now, but just wrapping around the nucleosome is not enough. So when you have a section of 8, so this is not quite a nucleosome, but 8 histone DNA complexes together, they form an octamer, which is called a nucleosome. All right, so first you have your linear DNA. You have linear DNA wrapped around a histone, which makes chromatin, right? It compacts it one way, and then 8 sets of histone DNA balls, really, clumped up together, make a nucleosome. Okay, so this reduces that 2 meter stretch of DNA till about 10 nanometers, so 10 times smaller than the size of a cell, right? And so now it can fit in the nucleus. Let's see, what else did I want to say about this figure? I think that's it. So we can see, once all of the DNA is compacted around the histones into the nucleosomes, that's how we get the creation of this X structure. So these are all bundles of nucleosomes formed up, and we can see them in this karyotype as homologous chromosomes here. Unlike the simplicity of binary fission, eukaryotic cells must undergo a more complicated division process because the eukaryotic genome is more complex. The cell cycle is an ordered series of events involving cell growth and cell division that produces 2 new daughter cells. So we talked about one type of cell cycle, which was binary fission. And now we're going to talk about another type of cell cycle for eukaryotic cells. And in eukaryotic cell cycle, there are two main phases, interphase, which we see here, and mitosis, which we see here. Actually, this whole thing is interphase. Some of the pictures cut off so you can't see. And then this little orange part is mitosis. During interphase, the cell is growing, DNA is replicating, and then during the mitosis, the replicated DNA and cytoplasmic contents are separated to prepare to go from one parent cell to two daughter cells. And then there's a subphase, so not one of the major phases, a smaller one, where the cell actually separates into two, called cytokinesis. Okay. So although the cell cycle is continuous, you can see it here in this circle, right? It's continuous and ongoing. I always think of the beginning of the cell cycle as G1. So I'm going to teach it to you that way. G1 stands for the first gap. It's a growth phase, and this phase allows newborn daughter cells time to basically mature. So during G1, the cell is accumulating the building blocks of chromosomal DNA, any associated proteins, as well as spacing out the organelles and accumulating sufficient energy to complete the task of replication all over again. So after G1, we can follow the arrows, the cell moves into S phase. During S phase, the DNA is replicated. So S stands for synthesis. DNA is replicated, which means a new identical copy of the DNA is synthesized in preparation for the creation of a new daughter cell. During S phase, the mitotic spindle is developed. Write this down now because it's going to become important during one of your assessments. So during S phase, the mitotic spindle is developed and chromatids are replicated to create sister chromatids and the chromosomes that form that lovely X shape that we're used to seeing. So after a cell, as the cell enters G1, there's only single chromatids. During S, replication happens and new sister chromatids, which can combine into that X shape, are formed. Once S phase is complete, the cell following the arrows moves into G2 or the second gap. During G2, the cell size adjusts to accommodate the doubled amount of DNA. Organelle replication starts to happen and the cytoskeleton breaks down in preparation for cell division. Now finally, at the end of G2, the cell is ready for mitosis. Mitosis is a multi-step process during which the duplicated chromosomes are aligned or lined up, separated, and moved into new identical daughter cells. Mitosis then ends, like I said, in cytokinesis, which is the physical separation of the cytoplasm, going from one parent cell to two daughter cells. I just want to point out some structures here that will become important as we go. So we can see the chromosomes here, the sister chromatids together in their traditional X chromosome structure. This is the nuclear envelope. You can see it's starting to break down. So this cell is getting ready to go into cell division. Oh, here are our chromosomes. These little structures here, these blue structures, are called spindle fibers. Okay, so these are made out of tubulin, and they are made from these microtubules, and then they are kind of organized by these centrosomes, which are these orange pieces here. These centrosomes direct the poles of the cell, so they tell the cell which is basically which side is east and which side is west, so that when division occurs, it knows where it's set, which parts is it pulling towards to create the two new daughter cells. All right, so that concludes the mini lecture on the cell cycle and mitosis. So now you're going to move on to the mitosis learning activity. Here's a hint. If while you're going through the mitosis learning activity, you draw out the steps of mitosis that you see there, then essentially you will have your somatic cell division review assignment, which is the next assignment after that. You'll have that done, really. So that's my advice. Draw out all the steps that you learned during the mitosis learning activity, and then use them for the somatic cell division review assignment. Okay, see you next time.